War and Medicine.
The Korean War broke out on 25th of June 1950, quickly disrupted the South Korean medical system, which has been hard-won under the US military regime and the newly established South Korean government. Thousands of hospitals were destroyed or closed across the country, and equipment and drugs were lost. But the war also brought about another change in medicine. The UN forces included a large number of military medical personnel who worked alongside South Korean doctors to treat wounded soldiers and contribute to community health. This naturally exposed South Korean medical personnel to the latest medicine and technology outside Korea. Advancements in surgery, including orthopedics, cardiothoracics, neurosurgery, and anesthesiology, were particularly notable. On the one hand, the medical personnel were heavily exposed to US medicine, which promoted Americanization. There were also changes in medical system as a whole. There was a need to provide health service to ensure the safety of huge refugee population and to stabilize the rear. Set up to provide a service needed to relieve refugees, health clinics provide the basic health service such as simple medical care and vaccinations. While not as stable as health centers, 471 health clinics was established by 1953. These temporary health clinics were later reduced and consolidated to form the basis of Korea's health center network in the 1960s.
DDT and Koreans.
In addition to the medical care received through the health clinics, new medical technologies developed during the war also captured the public's imagination. DDT was a prime example. Massive amounts of DDT were sprayed during the Korean War to control infectious diseases, to the point where one researcher described the DDTification of the Korean peninsula as occurring during the Korean War. Another thing to keep in mind is that DDT was first sprayed by the US military for disease control purpose in 1943. So in 1945 Korea, it was a new technology that has only been in the field for a little over 2 years. It was the first synthetic pesticide in history, rather than from the plant or inorganic raw materials. And it was also the first time in history that pesticides would play a significant role in disease control. The technology of organochloride insecticides like DDT and knowledge of their use and consequences remained rudimentary until the 1950s. In that sense, Korea, where DDT was used extensively for disease prevention in civilian rather than military applications, was an ideal test subject. Much of the US military stockpile of DDT from World War II was used extensively in US-controlled territories such as Korea and Japan, immediately after the end of the war. The major diseases in Korea and Japan then was malaria and typhus, which were seen as diseases that needed immediate intervention, especially as they could be transmitted to American soldiers. Each was an insect-borne disease carried by mosquitoes and body lice. So DDT as an insecticide soon became a key public health tool. After the war, returning Japanese and Koreans were required to be sprayed with DDT as part of immigration process during their return home. DDT was white talcum powder mixed with an insecticide solution, and it was only worked through direct contact, meaning that in the case of body lice, the insecticides had to be applied directly to the body to be effective. photographs of refugees covered in white powder are not uncommon. This exposure to medical technology became part of people's daily lives through the Korean War. On the one hand, DDT became an integral part of Korean life, as it was used as a pesticide to increase agricultural productivity. However, in November 1950, at the height of the war, reports began to emerge from the prisoner of the war camp in Busan that body lice did not seem to die in spite of large amount of DDT being sprayed. At first, the US military thought it was a problem of a spoiled old stock of DDT. But the problem was not with the older stocks. In response, in January 1951, the US military decided to increase the concentration of DDT powder used in a camp from 5% to 10%. But even with the higher concentration, DDT spraying in Korean POW camp had no effect on body lice within 24 hours. The conclusion was clear. DDT's effectiveness against body lice was rapidly diminishing. So much DDT has been sprayed on the Korean peninsula in such a short period of time that powerful evolutionary pressure were at work, and insect resistance to DDT were rapidly emerging. In 1950, 3,432,000 people were sprayed with 10% DDT powder in POW camps alone, and 134 tons of DDT powder was used. US military researchers formed a research network linking the US, Japan, and South Korea. The ultimate goal was to obtain insecticide-resistant body lice, found in Korea, and measure the extent of their resistance. Until the 1940s, the understanding of insecticide resistance was that there were certain individuals or species that were particularly hardy. They may have thick shells that neutralize the effect of contact poison, or they had unique behaviors that allow them to avoid insecticides. In other words, the prevailing view was that insecticide resistance did not emerge as a result of external evolutionary pressure from insecticide application, whereby individuals were selected for resistance, but rather as a result of inherent trait or behaviors that the insect species had in the first place. According to this understanding, it was sufficient to change the formulation of the insecticides, of the way it was applied to the evade resistant. However, the resistant body lice found in Korea did not look or behave significantly different from susceptible ones, meaning that they had acquired insecticide resistance based on separate physiological mechanism, rather than behavioral or physical aspect. At the same time, it also meant that if insect resistance was physiologically inherent in insect, change the formulation of insecticides or the way it was applied would no longer be effective. The discovery of DDT-resistant body lice in Korea in 1951 changed the way we think about and understand insect resistance. It showed that insects could acquire resistance to synthetic insecticides through evolution quickly, and that insecticides like DDT were not magic bullet to solve all problems. Scientific methods for breeding insects in the lab and exposing them to pesticides to measure resistance were developed and standardized. Methods for mixing multiple pesticides were devised to reduce the likelihood of insects acquiring resistance. The Korean body lice then traveled to the United States, where they reinvigorated insecticide resistance research. However, the way in which this knowledge and research resource were collected and utilized by US military researchers in Korea need to be viewed critically. Korean body lice were collected from Korean volunteers, and experiments that might have caused discomfort to the subject, such as those involving direct body lice attachment, were conducted on Korean soldiers or POWs, not US soldiers. It is worth rethinking the role of public, soldiers, and prisoners in the development of medical research during the Korean War, especially in light of criticism that advances in medical knowledge have relied on experimentation and observation on marginalized populations.
Side effects of the medical technology.
Despite the problems of resistance, DDT changed the people's lives dramatically. DDT as a household insecticide, pest that has been reducing people's quality of life, were quickly flushed out. Disease vectors such as mosquitoes and body lice were also greatly reduced. And it became commonplace to use aeroplanes to spray pesticides such as DDT over large area, as shown in the photograph. The sight of military planes flying overhead and spraying pesticides evoke images of the war against the pest, a carpet bombing campaign. While aerial spraying did little to actually reduce household pests, it created a visual illusion that the government was conducting an effective control program. Local politicians lobbied the United States Air Force to conduct aerial spraying in their cities. As the case of DDT shows, new medical technologies, such as insecticides, have penetrated deeply into people's daily lives. In particular, insecticides have been widely utilized as an important tool to increase agricultural productivity. However, as with any new technology, there are both positive and negative aspects. Here, we'll explore how new public health tools can have both sides of the coin, using the examples of the pesticide poisoning. The Green Revolution in agriculture since 1950s has dramatically increased the food production in low and middle-income countries. But it has also created new health and ecological problems by increasing the use of pesticides and fertilizers. The overabundance of pests caused by intensive agriculture has led to increased pesticide use, and with increased pesticide use comes increased evolutionary pressure, which has led to the rapid spread of insecticide resistance in the pest. As pests became more resistant, more toxic chemicals were used, and heavier doses of chemicals were used for quick fix, increase the frequency and amount of pesticide exposure for the farmers who spray them. This widespread use of pesticides in 1950s has unforeseen consequences, one of which was the problem of pesticide poisoning. Even today, there are about 3 million cases of pesticide poisoning worldwide annually, and it is estimated that 14 million people died from pesticide suicide worldwide between the Green Revolution in 1950s and 2018. In South Korea, pesticide poisoning as well as pesticide suicide was a significant health issue until early 21st century. As mentioned earlier, DDT was begun to widely used in agriculture during the Korean War, and just like in the public health sector. Agricultural pests quickly began to develop resistance to it. A new pesticide that emerged was parathion. Unlike organochloride pesticides such as DDT, which were primarily effective only through direct contact or ingestion, organophosphorus pesticides, that is parathion, were penetrative, meaning they could penetrate into the plant's tissue and spread up the stem and outwards, affecting pests that were parasitizing within the plant, not only living outside. However, this potent toxicity and penetrative nature also apply to animals, including humans. Parathion inactivate the enzymes responsible for breaking down neurotransmitters, causing the nerve system to become excited, which in turn leads to the convulsion, coma, respiratory failure, and eventually cardiac arrest. Comparing the toxicity of the DDT and parathion, parathion is approximately 70 times more toxic than DDT when ingested orally, and 370 times more toxic when absorbed through the skin. Parathion was first imported into South Korea in 1955 through pesticide import funding from the U.S. International Cooperation Administration, ICA. U.S. aid officials hoped that the chemicalization and mechanization of South Korean agriculture would increase production. To increase rural pesticide use, U.S. distributed 20,000 posters in December 1955, encouraging farmers to aggressively use pesticides if their crops show signs of disease. Following the change in the aid policy, the supply of pesticides to rural South Korea began to increase rapidly. Total retail sales of pesticides rose from about 770,000 liters in 1953 to 1 million liters in 1954, and 4 million liters in 1955, and 6.7 million liters in 1957. On 3rd of August 1956, a series of sudden deaths of children occurring in North Gyeongsang Province, southern part of Korea, where it was famous for apples and fruits. Initially, it was assumed to be an infectious disease of unknown origin. However, epidemiological investigation concluded that the characteristics of the death were close to poisoning rather than infectious disease. It was the head of health clinic working in the community who revealed the identity of the disease. Having cared for patients from the beginning of the outbreak, he realized that the clinical symptoms were similar to poisoning, and while reviewing the literature, he found a case of parathion poisoning in a Japanese pesticides book, and realized that the symptoms were identical to those he had seen in Korea. Once it was known that the cause of outbreak was pesticides, it was pointed out that the importation, sale, and use of high-risk pesticides such as parathion was indiscriminate. The government first enacted the Pesticides Control Act in August 1957. In light of the poisoning incidents caused by the highly toxic pesticides being sold as general household pesticides, it was mandatory for pesticides to be labeled as pesticides on their containers or packaging and to list their specific ingredients and precautions. Pesticides were a major technological tools that increased the agricultural production in post-liberation Korea, but they were also created new public health problems. Japan, which was a key reference point for South Korea's pesticides management legislation, attempted to manage highly toxic pesticides such as parathion in separate notifications. But these follow-up measures were rarely reflected in South Korean legislation. Paradoxically, parathion's high toxicity was imprinted in public's mind, and it was used as means of suicide, creating another health problem.